Top Med Talk.
Hello, I'm Joff Lacey and welcome to Top Med Talk, and this our very special series of podcasts on Xtreme Everest, the groundbreaking research program that for the past decade now has pursued a novel approach to scientific exploration, performing large-scale healthy volunteer studies at high altitude to mimic the effects of critical illness, with the hope that their findings may be brought from the mountainside to the bedside and improve the care of our sickest patients. In these podcasts, we will take you through the story of Xtreme Everest from its inception to its discoveries and to the future of this unique endeavour. For this episode, I have the pleasure of being joined by Dr. Denny Levett, a consultant in intensive care and perioperative medicine at the University of Southampton, and one of the original founding members of the Xtreme Everest group. Welcome, Denny. Thanks for joining me.
Thank you very much for inviting me along.
Pleasure. So, Denny, Xtreme Everest has a mission, I would say, in trying to unravel the secrets and misunderstandings of how the body responds to hypoxia. Can you just tell us what is hypoxia?
So hypoxia refers to the condition where not enough oxygen is getting into your body and to your cells. And oxygen is the really important fuel that, along with food, allows us to produce all the energy that we produce to do everything that we do: move, grow, reproduce. And we need a constant supply of oxygen from the environment to our cells to be able to perform normally. And when those levels drop, the condition is referred to as hypoxia. And that can be caused, firstly, because there may not be enough oxygen in the environment, but it can actually be caused equally by a problem anywhere along the delivery chain from the outside air to the cell. So it can be caused by having lungs that are working badly, for example, in pneumonia, or if your heart isn't pumping the oxygen effectively around the body in heart failure or after a heart attack.
And so you work in intensive care, you look after very sick patients. Where does hypoxia fit into looking after those patients? How common is it?
So the final common mechanism of injury in critically ill patients in intensive care is usually hypoxia. Now, the causes of that are myriad in patients. As I said, the oxygen supply can be interrupted at any point in the chain. In fact, if you look in intensive care patients, over 90% of them will be hypoxic at some time during their critical illness. So it can be because of a problem with lung function, problem with heart function, the peripheral circulation can be affected, or sometimes actually the cells themselves can't use the oxygen that's available.
So despite all these patients having a multitude of very different pathologies and problems with their bodies, they're all kind of unified to some degree by this condition of hypoxia.
Absolutely. So there's a myriad of different medical problems that you can have, a long list of things that result in the mechanism by which you cause injury to your body, and in fact, sadly, in many cases, you die in intensive care is because we fail to deliver oxygen to the cells or use oxygen.
Some people listening will just say, well, why don't you just give more oxygen? Is that not just the quick fix? Why is there a need for the scientific inquiry? What are the problems? What don't we know about it?
Well, there are a number of things that we don't understand about how the body responds to low oxygen levels. One of the things that's really noticeable as an intensive care doctor is that some individuals are able to tolerate oxygen deprivation much more effectively than others. And it would really help us to understand how to treat patients better if we could identify those patients who responded poorly and got organ damage very early in their critical illness in comparison to others who can tolerate low levels of oxygen better. And the reason that's important is that oxygen is a double-edged sword, essentially. It's obviously beneficial and it's necessary for all cells, but too much oxygen, in the same way that too little oxygen can be dangerous, can actually cause damage. And not only that, some of the things that we have to do in intensive care in order to try and get oxygen through, for example, damaged lungs, means that we can actually harm the patients inadvertently. So the way we breathe naturally is very different to the way that we mimic breathing in intensive care with life support machines and ventilators. And sometimes by using those ventilators and giving high levels of oxygen, we can actually counterintuitively damage the lungs at the same time. So it would be very useful to know how little oxygen we can survive with, and to identify which patients need more oxygen or less oxygen when they're extremely unwell.
And you mentioned about some people seemingly doing better than others with the same kind of physiological hit of hypoxia. in terms of working out who can fare better than others, it's not a straightforward process. It's not simply that if you're fitter, you can deal with it. Is that correct?
Yeah, no, that's very correct. There isn't a single factor that we understand at the moment, certainly, that allows us to predict who's going to tolerate lower levels of oxygen better. It's not simply a matter of how much oxygen you can deliver to the cells. It's also about how your cells use the oxygen that they actually receive. And individual variation between different patients or between individuals probably comes down to differences at a cellular or metabolic level in terms of how we use that oxygen.
So we don't completely understand how the body responds to it. We don't understand how some people fare better than others, and potentially some of the seemingly aggressive interventions we're doing may be doing more harm than good.
Absolutely. And then finally, if we could identify the characteristics of the sort of star hypoxia performer, we would hope in the long term, in the future, perhaps be able to manipulate the pathways that are in their cells that make them such good adapters to hypoxia, so we could develop treatments for those patients that we know don't tolerate hypoxia so well. In the future, that's our real main aim, that we can identify poor hypoxia adapters and have targeted treatments for those individuals.
So if we could move on to the actual Xtreme Everest project itself, tell me where the idea came from. Why take healthy people up to Everest to look into ways of trying to improve the care of these intensive care patients? Some people might seem slightly incongruous.
So there are a number of challenges to doing work in an intensive care environment. Firstly, the patients in intensive care have a wide variety of underlying conditions. And although, as I said, they often have the common feature of hypoxia, it's one feature that's mixed up with the background of what their underlying condition is. And so it becomes very difficult to carefully study what is the effect of the hypoxia itself versus what's the effect of the underlying condition they have, whether they've had a heart attack, whether they've got meningitis, whether they've just had major surgery. So actually to tease out the effects of hypoxia within the critical care environment is difficult. Secondly, one of the other challenges in intensive care, ethically in terms of research studies, is patients are often unconscious and sedated because they are so unwell, which it makes it difficult to obtain consent to do complex studies. And finally, we felt it's very important to look at the effect of hypoxia within the whole body. There are a number of experiments that you can do looking at cell preparations or at animal models, but unfortunately, particularly in critical care, in fact, we've had a long track record of treatments that have worked well in animal models, but when that's been translated to the patient environment in critical care, they've been shown to be not effective. And hypoxia is a condition that affects all of our organs at once, and so actually understanding how the whole body responds rather than a single cell is really important. And so by taking healthy individuals and exposing them to different levels of hypoxia, it allows us to look at the effect of hypoxia itself rather than the underlying condition, and we can do that in the whole organism, as it were. And by taking healthy individuals to their limits, we can see what the limits are to tolerating hypoxia, and as I said, look at enough individuals to identify the ones that perform particularly well.
So the first Xtreme Everest expedition was just over 10 years ago now, 2007. Could you kind of take me through the design, how it worked? What was the kind of structure to the expedition?
Sure. So because our aim was to help patients back in critical care, what we were not doing on Everest was looking at altitude illness. What we wanted to do was take a large group of individuals and look at variability in how they performed. Many previous altitude studies have been in small groups of climbers who are actually probably self-selecting as good hypoxia performers. So we took 200 general public volunteers and we exposed them to hypoxia in a standardized way by making them ascend at a standardized rate to altitude. And as you climb at altitude, because of the changes in environmental pressure, the amount of oxygen that's available to your body decreases. And so by the time an individual gets to Everest Base Camp, there's about half the oxygen available that there would be whilst we're sitting in this room. And so by making these 200 general public volunteers ascend at exactly the same rate, we could do essentially a dose study of hypoxia. And we tested them all at baseline, and then we tested them at 4 different altitudes in Nepal to evaluate how they changed as they became exposed to greater and greater levels of hypoxia.
And what kind of tests were these subjects undertaking?
So we were very keen to look at really hypoxia's effects on all the organs. So there were a multitude of tests during which we evaluated brain function, cardiac function, our ability to exercise, how the lungs were functioning. So those are sort of large whole body physiology tests. And then we also looked at what we call the microcirculation, which is the tiny little blood vessels which are responsible for actually delivering oxygen to cells and tissues. So we looked at the small blood vessels under the tongue in particularly, and also affecting the stomach. And then we looked at how the cells use oxygen by taking muscle samples and looking at the mitochondria and how they were functioning both at sea level with normal oxygen levels and when the volunteers were hypoxic. And as well as the general public volunteers, which were these 200 individuals that went to Everest Base Camp, we were keen to investigate the extremes of hypoxia tolerance. So a smaller group of investigators, who were all doctors as well, then climbed on Everest to higher altitudes, so greater degrees of hypoxia, to look at what limits performance at extreme levels of low oxygen levels, essentially. And we also evaluated with them cognitive functions, so how the brain works in hypoxia, and did the more invasive studies where we looked at taking muscle biopsies from their legs.
You mentioned some of the team went all the way to the summit, and one of the members, Dr. Dan Martin, has a dubious claim to fame regarding some of the analysis of the blood tests they did just below the summit. Can you take me through that?
Yeah, so one of the things we wanted to look at was after a period of acclimatization, which is a period where you're exposed to low oxygen levels, actually we tolerate them much better than if we expose a patient to very low oxygen levels immediately. So if I took you and put you on the summit of Everest right now, you'd be unconscious within about a minute. But our climbers had 6 to 8 weeks to get used to these low levels of oxygen. And what we did was take blood samples from the artery in the leg so that we could see how low the levels were at the extremes of altitude. We planned to take that blood gas actually on the summit itself. But on the summit day, it was extremely cold and windy, so the decision was made to come down about 200 metres from the summit. And we took these blood samples from the groin, and Dan's sample was the lowest oxygen level ever recorded in a conscious human. So I think people were surprised at the degree of hypoxia.
And so a real illustration of how the body can adapt, because not only was he alive and conscious, but he was functioning to a reasonably high level.
Absolutely. It just goes to show that given time we can adapt to quite severe degrees of hypoxia, but that's the other feature that's quite important. It's not just how low the oxygen levels are, but it's how long you've had to get used to that degree of low oxygen levels.
And that would be an important thing, because a lot of intensive care patients don't have a nice graduated exposure to this insult. It can be quite sudden.
Yeah.
The idea being that somehow if you can work out the mechanisms that allowed Dan to function like he did at the summit of Everest, if you can then manipulate that and allow patients to suddenly be able to have that adaptation and tolerate those low levels of oxygen, you won't have to go through all the aggressive, invasive interventions that may actually be doing more harm than good.
Absolutely. So that if we could move patients into the sort of state you see after acclimatization, where you're tolerating low oxygen levels much more quickly, we could avoid giving them large amounts of oxygen, which, as I said, can actually cause some harm.
That was your first expedition. You must have had an enormous amount of data.
Yeah.
What were the kind of take-home messages?
The first and important take-home message is that just measuring the amount of oxygen that's carried in your large blood vessels doesn't predict how someone performs. So your performance at altitude is not related to the amount of oxygen circulating in the large blood vessels. And in fact, it seems that the adaptations that happen in the really good altitude performers happen either in the tiny blood vessels or in the cells themselves. And that was probably the big take-home message from Everest. What we found was that overall performance at altitude didn't correlate with the amount of oxygen we're delivering from the heart and that leaves the heart. But when you looked down in the tissues, we found that the mitochondria changed and their efficiency changed in those individuals that were performing best at altitude, and also the microcirculation, which is the tiny blood vessels near the cells. The function of the microcirculation also changed very significantly.
Now, that's quite a game changer, because current medical practice focuses on the amount of oxygen that's carried in the major blood vessels and how much is being pumped round and round. But actually you found that shouldn't really, I mean, it's obviously important, but that's not the be-all and end-all.
It's not the be-all and end-all, and I think we actually are aware of that from our outcomes in critical care when we see these people that don't perform so well, even though we've normalised the amount of oxygen in the main blood vessels, because probably what's happening is that there is a blockage somewhere between the main blood vessels and actually the mitochondria, which are the little powerhouses in the cells where the oxygen is finally used to produce energy. But that's been a very important finding, and over the last 10 years since we published our data, along with other data that's come out of more traditional clinical studies, it's becoming clearer that actually giving large amounts of oxygen may not be the answer. And in fact, we are now just embarking upon a study with Dan Martin and our team in Southampton as well, looking at trying to restrict the amount of oxygen we give in critical illness to try and avoid causing harm by limiting and targeting lower oxygen levels, which hasn't been traditionally what we've done. And our work certainly on Everest has helped contribute to the sort of sea change in attitude towards more of a focus on the harmful effects as well as the beneficial effects of oxygen.
So the team then went back to Nepal and to Everest in 2013 on Xtreme Everest 2. How did that expedition differ from the first one? What were you looking at more specifically there?
Well, as always in research, where you're going to find answers, you get some surprises, and then that usually leads to more questions rather than complete answers. So on the basis of what we found in 2007, there are a number of things we wanted to look at in more detail in 2013 in Lowlanders, and that was, to summarise those, number one was to look at mitochondrial function in a bit more detail, and we wanted to look at it on fresh muscle samples rather than frozen ones, because there's different techniques you can do.
That sounds a pleasant sampling process.
Yes, exactly. And secondly, we wanted to look harder at the microcirculation, those tiny vessels, and what is it that's determining the flow in those vessels. And one of the important chemicals appears to be something related to something called nitric oxide, so we wanted to do some research looking in more detail at that, because those seem to be the two key areas. Another interesting finding from the first expedition was that people that had been to altitude on multiple occasions previously, namely our summit and investigator teams, appeared to be different at baseline from some of our other subjects who were healthy volunteers who'd not been to altitude before. That raised some really interesting questions as to whether it's because the summit team has selected themselves because they are good adapters and they have a different genetic profile.
And different in what respect? As in,
so they appeared to perform better, and they had different mechanisms of oxygen utilisation at baseline.
Right.
So when we looked at them in terms of the cardiac muscle functioning and skeletal muscle functioning at baseline appeared to be different. And so one of the things we want to look at, that may be that they've inherited a profile that makes them perform better, but one of the other areas we were curious about was it because they have some memory from previous exposures?
Yeah.
And we know, for example, that premature babies who've been exposed to hypoxia immediately after birth have a different response later in life when they're exposed to hypoxia subsequently. An evolving area of research is the field of epigenetics, which is how essentially your genetic information is altered by your environmental exposure, and it's a very nascent area in hypoxia research. But this interesting finding was something we wanted to explore more, so we looked at some people who'd been previously and looked to see how their patterns of response changed on a second time. And we also looked at some identical twins in 2013 to look at differences between genetic change and epigenetic change.
And then you also, interestingly, focused in on probably the best adapted people in the world for performing in hypoxic conditions, the Sherpa people.
Absolutely. So that was what I was talking about there, was in the lowlanders, and by that I mean people that have lived at generations at sea level. And we also wanted to identify what it was in the biology of the high altitude natives, so the Sherpa population, to see what it was about them that makes them such supremely good performers in this environment. And I think anybody who spent any time at altitude appreciates how much easier they seem to find it than us, and even if they are relatively slight individuals, they seem to be able to carry huge loads high on the mountain at a much greater speed than us. So that was interesting to us as well, to look at that in more detail. And having formed very good relationships with our Sherpa teams in 2007, we felt that we would be able to go back and do some studies to try and tease out what it is about the Sherpas that made them perform so well.
Because a bit like what you've already alluded to, it was known that the Sherpas don't exhibit what you consider to be the kind of traditional, so to speak, understanding of how someone should adapt to low levels of oxygen, so that they don't have higher levels of oxygen coming around the body, they don't pump more blood round and round, there's something else going on.
Yeah, so the traditional story of how we adapt to low levels of oxygen is that things in our body change in order to try and transport more oxygen. So we breathe faster to get more oxygen in, we make more red blood cells to carry more oxygen around, and it was felt that those individuals that had the biggest response in breathing rate, in levels of hemoglobin, might perform the best. But what we've found in Sherpas is almost the opposite. They don't particularly have high levels of oxygen saturation, so they don't breathe much faster than the lowland population. And in fact, if you look at their hemoglobin levels after long periods of exposure to very high altitudes, they are much lower than the equivalent lowlander population. So it seems that they are performing extremely well, but their performance is not explained by increasing the bulk amount of oxygen that they're getting from the environment into the large blood vessels. And so on them, given our findings from 2007, we wanted particularly to focus on, again, the small blood vessels, because there was some suggestion from some other research groups that they had very high levels of this nitric oxide chemical, which might be affecting the flow to the microcirculation. And also we were interested to see if their mitochondria were such that they use their oxygen more efficiently than us.
So we're going to be talking to some more members of the Xtreme Everest team about those specifics, the microcirculation and the mitochondria later, so I won't spoil it for listeners. But I will ask you, so with all the information you've had, and still, in a way, so many questions, is the team going back? Is there a plan for more work?
I'm sure we will go back at some point. There isn't a date set just yet, and the reason for that is actually we are dealing with a mountain of data, as it were, right now. It's a massive organisational challenge to put these large field expeditions together, and I think it's really important to be clear about what your focus questions are. So there is a little bit of work, particularly at the molecular level, that we are still working our way through at the moment. So I think not in the next couple of years, but in the next five-year frame, we may be going back to ask more questions. There'll certainly be more questions to answer. I think it's not a simple question and there's going to be one answer to.
Look, Denny, I have to leave it there. We're out of time, but thank you so much for joining us today. Please visit the Top Med Talk website on www.topmedtalk.com for the rest of the podcasts in this series, where we'll delve further into the discoveries of the Xtreme Everest team. And there's plenty of embedded related material there to further whet your appetite. And that's all from me for now. Many thanks for listening.
Top Med Talk.